Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will continue with the biological buffers, the ammonia buffer system, which plays an important role in the kidneys and helps our body excrete excess acid through urine. Let's understand how it is. Let's understand what is the ammonia buffer system. Before that, in general, we know that what is actually a buffer. What is actually a buffer? It is a combination of a weak acid and its conjugate base or weak base and its conjugate acid. What is the function of the buffer? Actually, why we are taking buffer in all the chemical reactions or even in the body. So actually, what is the function of the buffer? It resists pH changes. What actually? It resists pH changes in the body. Now we are talking about ammonia buffer system. So here, if the pH is very low, the buffer will make it to normal. If the pH change is very high, again the buffer make it to normal. Means here the buffer makes the pH changes to normal or stable. This is the function of the buffer. Now let's see what is the ammonium buffer system. So here ammonium buffer system, actually it primarily functions in the kidneys, especially in the renal tubules. Now what are the components of the buffer system? Means ammonia buffer system. So here it is ammonia and ammonium ion. What is ammonia? Here ammonia acts as a weak base and ammonium ion acts as its conjugate acid. Can you tell me how exactly ammonia buffer system differs from other buffer system? If you know the information, please comment me. Okay. So here in most buffer systems, usually we see the combination of weak acid and its conjugate base, isn't it? Right? For example, if you see in bicarbonate buffer system, the components of buffer system are carbonic acid, H2CO3, it is a weak acid, and bicarbonate ion HCO3 minus, so it is acting as its conjugate base. And if you see in second case, phosphate buffer system, so in phosphate buffer system, what are the components of the buffer? One is dihydrogen phosphate, H2PO4 minus. So here it is acting as a weak acid, and monohydrogen phosphate, HPO4 2 minus. It is acting as its conjugate base. So these are the combinations we have seen in the case of bicarbonate and phosphate. Now in ammonia buffer system, what is the combination? So here we will see the combination of weak base and its conjugate acid. So here, what is acting as a weak base? Ammonia. So here, ammonia acts as a weak base, and ammonium ion acts as its conjugate acid. So what is the function of the ammonia buffer system? Actually, the function of the ammonia buffer system is to get rid of excess acid from the body, right? So here, what is extra acid? Extra acid is nothing but extra H plus ions. So these extra H plus ions are removed from the body by trapping as ammonium ion. By trapping them as ammonium ion, which is then excreted from the body through urine. Now let's see the whole mechanism. So here it takes place inside renal tubules. So what is the first step here? First, the glutamine enters kidney cells. Tubule cells, renal tubule cells or kidney cells, both are same only. So here, now, how is this glutamine produced? So here glutamine is actually a amino acid which is taken up by the kidney tubule cells from blood. So now this glutamine inside the kidney tubule cells will be broken down into ammonia and bicarbonate ion. So now this ammonia will goes into urine and the bicarbonate ion again enters into the blood. Now this process helps the body to gain extra base. Now here what is acting as the extra base? Bicarbonate ion. So here bicarbonate ion acts as an extra base to reduce acidity. First, we have seen that glutamine will convert into ammonia and bicarbonate. This is the first step. Second, the ammonia which has been produced from the glutamine. Now, this ammonia will diffuse into tubular lumen. Right? So, here ammonia naturally it is being a gas, it easily diffuses into the tubular lumen. Is it? So, third step, now H plus is secreted. Now this H plus is also secreted into the tubular lumen. Now let's see how the H plus is secreted into the tubular lumen. So first, we have already know that carbon dioxide is produced during metabolism, right? Now this carbon dioxide reacts with water in the blood plasma to form carbonic acid, and from there again it is converted into H plus and bicarbonate ion, right? Now this bicarbonate ion enters into the blood. Now this H plus will secreted into the tubular lumen. Is it clear how it has been secreted into the tubular lumen? So finally the ammonia which has been formed from glutamine and the H plus from the carbon dioxide now will convert into ammonium ion. So here ammonia will bind to H plus to form ammonium ion. So here, what is this H plus again? It is excess acid. So this ammonia will neutralize the excess acid, forming ammonium ion, which is then excreted from the body through urine. So why this ammonium ion is excreted from the body through urine? Because it cannot cross cell membranes. Because it cannot cross cell membranes. As it cannot cross cell membranes, it will be trapped in the urine only. So from there it is excreted from the body through the urine. So from this it removes excess acid from the body, right? So here excess acid has been removed from the body through the urine in the form of ammonium ion. So meanwhile, the bicarbonate ion which has been produced from the glutamine first pathway and the second from the carbon dioxide, both will enter into the blood in order to neutralize acidity. So is it clear, the whole mechanism? Now let's see what is the importance of ammonia buffer system. Actually, first it handles large amounts of acid. Second, it can help the body to maintain acid-base balance in the long term. Third, it is specially active when other buffer systems like bicarbonate and phosphate are not enough. And finally, unlike fast acting buffer, what is fast acting buffer? So here fast acting buffer is the bicarbonate buffer. Unlike fast acting buffer, ammonia buffer also works slowly but very powerfully in the long term. Now let's quickly revise what we have read. Uh, First, the ammonia buffer system operates in the kidneys. Second, it removes excess acid from the body through urine. So here ammonia combines with extra H plus to form ammonium ion, which has been trapped in the urine and it is excreted from the body through the urine. And finally, it is specially active when there is a chronic acidosis or other buffer systems, or if other buffer systems are insufficient. Is it clear? That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the video, please comment on your doubts. And if you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. Please do support me. And in the next video, we'll cover the topic Henderson and Hasselbalch equation and the related problems. So until then, keep learning and stay curious. See you in the next class.